Water regulation in leaves is vital to a plant's health, affecting its growth and yield, disease susceptibility, and drought resistance. A breakthrough technology developed by Cornell University researchers uses nanoscale sensors and fiber optics to measure water status just inside a leaf surface, where water in plants is most actively managed. The engineering feat provides a minimally invasive research tool that will greatly advance the understanding of basic plant biology and opens the door for breeding more drought-resistant crops. The technology could eventually be adapted for use as an agronomic tool for measuring water status in crops in real time. The study in maize plants, a minimally disruptive method for measuring water potential in planta using hydrogel nanoreporters, is published in the journal Proceedings of the National Academy of Sciences. Current techniques for measurements of water potential require destructive sampling of leaves or disrupting leaf function. The new method provides minimally disruptive and spatially and temporally resolved measurements of water potential in leaves of intact plants. Outside of leaves' transport tissues called the xylem lies an interior zone called the mesophyll, where most of the plant's photosynthesis and water stress occurs. Biologists suspect that signals are sent from here to the rest of the plant for managing water. Also, at the surfaces of leaves and stems, pores called stomata open and close to control the rate of exchange of gases, mostly water vapor and carbon dioxide. The new technology works in this microscopic zone. The technique involves injecting nanoparticles formed of a soft synthetic hydrogel called echo dust for measuring a leaf's water potential. The hydrogel, which occupies the interstitial spaces between cells in the mesophyll, is water absorbent, swelling and shrinking based on water availability in the leaf. The echo dust contains dyes whose interactions allow it to fluoresce at different wavelengths depending on how close the dye molecules are to each other. By using fiber optics, the researchers can shine a light and get a spectrum back, which provides a measurement of water potential inside the leaf. In the study, the researchers injected the echo dust in multiple places along meters-long maize leaves and then measured the water gradients both along the length of the leaves and through the mesophyll. These measurements allowed them to develop a model of the tissue response to water stress and accurately predict the dynamics observed in the field. This technology may have commercial applications for crop research, production agriculture, and manufacturing industries. But for now, the researchers' focus is on the invaluable measurements of very local physiology of water management in plants. As a research tool, it allows plant biologists to better understand extremes of water stress, which could lead to breeding more water efficient crops.